Hi everyone. Today I would teach you the fundamentals of uh, QSAR, that is especially with pharmacophore modeling, uh, that is e-pharmacophore design, and how one will start with an e-pharmacophore based drug design and how it will fetch you to improve your research in pharmacophore modeling. Let us go back to some fundamental aspects of e-pharmacophore modeling. Simply, if you type e-pharmacophore guided research article, it's a very good research article in the recent past. It tells you regarding the e-pharmacophore guided 1-pyrazoloquinozolines as dual inhibitors of topoisomerase inhibitors and H1. There are some there is some what do you say, pattern of drug design is well defined in this research article. Now we are following this research article to do the current research and to perform the same kind of docking, especially the pharmacophore guided docking with reference to this article by using a software called as Discovery Studios. So I would like to thank the Discovery Studios for providing me the license to do research on COVID-19. So let us go back to the pharmacophore modeling by using Discovery Studios. So the ultimate pharmacophore would look like this, which you are seeing on my screen, which will tell you various descriptor parameters which are available in your ligand. Let us start from the basics, how you design a e-pharmacophore. For that, you just go for macromolecule. In that macromolecule, you just type the the protein of your interest. That is, now here I am just simply typing 1SC7, that is DNA topoisomerase 1, which is active for treatment of cancer, where a camptothecin analogs are bound in the active site along with the DNA templates. So here you are able to viewing the, what do you say, the fundamental aspects of the 1SC7 structure with a co-crystal ligand as well as the DNA. Now if you see, if you double click on this molecule, this is your ligand of your interest that is already bind inside the active pocket. This is your fundamental information which you are getting from the what do you say, active site of the 1SC7. You can simply go back to your PubChem database and simply type 1SC7 in the Google. Once you do that, it will go to DNA topoisomerase 1. So here I'll just go back to you and I'll show you the fundamental aspects of interaction of your ligand. If you see, it's clearly telling you about that isoquinoline molecule is present in my active pocket. If you see here, the isoquinoline is bound to DNA as well as certain residues inside the active pocket. So this is the fundamental information which you are getting out of this research. So let us go back to our software where we have downloaded the what do you see the protein the protein that is 1sc7, which is bound to DNA as well as the what do you say the active pocket inside the protein. Now simply you have to follow all the basic steps, that is protein minimization, simulation, protein preparation, and how to minimize the protein, how to get the perfect structure of the protein, which I already discussed in my earlier lecture. In the current lecture, I am just focusing on creation of e-pharmacophore model and screening the ligands onto the e-pharmacophore model. That's it. I'm not going to explain you what is protein, how you will quantify the protein, how you will minimize the protein, how you will find the active site. These are all things which I am not explaining in the current lecture. However, you can find them in my previous lectures, which are well available in my YouTube channel. Now here, whatever the ligand groups, if you are able to see, so those ligand groups, I am just simply collecting this ligand group. I am just cutting those ligand groups from the pocket, that is deleting them from the pocket, and I'll go to the file new and simply click new molecule window. In the new molecule window, I'll simply paste the ligands. So this is my ligand which is present in the active pocket of 1sc7. This is very simple. So you can even see some other molecules are there which are not essential for your what do you say active pocket docking. Such molecule you can remove, and only the active sited molecule, which is preferably your active molecule, you should always able to discriminate between the active pharmacophore as well as some amount of cofactors. So what are cofactors? These cofactors are added while a protein precipitation. So these things you should not confuse along with your active site of your ligand. So this is a fundamental information. So here, this is my active molecule. So again, I am not teaching you about the what do you say minimization of structure, how do you make the ADME analysis, everything which I have discussed in my earlier lecture. So here simply I am going to teach you the pharmacophore modeling. So simply I'll go back to this one. I'll click on 1sc7 and I'll go to the module called as pharmacophore. Once I go to the module of pharmacophore, here I have a module called create pharmacophore automatically. After selection creating auto pharmacophore molecule window, I got an interactive pharmacophore generation. Simply going to the interaction pharmacophore generation window where I have selected 1sc7. Now in this ligand portion, I will give the molecule which I have deleted from the active pocket. So let us select the molecule, that is this molecule 1sc7, which I have deleted from the active pocket and pasted in the new window. I am selecting and I'll simply run. Once I do that, what happens is this molecule fits into the active pocket and form a three-dimensional interaction with the receptor pocket and will tell you what sort of interaction which is having, whether it is having hydrogen bonded, whether it is having hydrophobic interaction, whether it is electrostatic interaction, whether it is having intramolecular bonding, whether it is having DNA chelation. So these are all appearing on your screen as peers. So that will appear after the completion of my job. The jobs can be viewed from the bottom window. Simply I'll click background and after that I'll go to jobs. In this job, you can see the interaction pharmacophore generation has been done. Simply I'll click OK. Once I click the completed job, I'll double click this So this is my interaction pharmacophores. There are around 10 pharmacophores generated. I'll simply click view results. Once I do that, my pharmacophore get displayed. So these are the pharmacophores which I have been designed for my active pocket of. You can see there is an active ligand also inside it, which is placed perfectly into the active pocket with certain amount of interactions. So let us see what exactly those are. Simply I am doing this, and here I'll go for receptor ligand interaction. Now I'll click this molecule, sorry, not this, this one, and I'll click define ligand, and I'll go for define this, and I'll go for define receptor, and I'll simply click ligand interactions. Once I do that, this is my ligand pocket which is having interactions 3-dimensionally. So this is my pharmacophore which I have generated. Once my pharmacophore is generated, now I should screen my pharmacophore with existing drugs as well as design drugs by my research. So let us do that. Simply I'll go to structure, and I'll go for label and add label. Once I do that, go for amino acid and simply put name. I'll click OK. Once I do that, you can see this arginine 364. This is exactly the active site. And if you just click the nucleic acid portion also, you can see the nucleic acid binding. You can be visualized. This is the fundamental part, and you can even click the proteins. So this is the protein which is having in the active pocket along with the nucleic acid. So this is the fundamental information which you are getting from the docking part. After doing this, you simply go back to your just simply hide the ligand from the active site. So now you have hide the ligand so that you are fitting the other ligands into your active pocket. So this is this difference between hiding and fitting. You should know that the active site should be remain blind so that the incoming molecule should not have any interference into the receptor pocket. Now let us screen. Already told you the research article suggested you so that after screening they got certain amount of molecules which have been designed by themselves in their laboratory, and they fit into the pharmacophore. This is the basic module which they have done. So this final molecule which they have designed, I have sketched and I kept on my desktop. That molecule I will be using in docking. So let us go back to the software, and simply that molecule I have named as Q. So this is the molecule which is the final molecule which they have been designed in the literature. So let us dock this into the QSAR model and see how well this fits into the QSAR model. For that, I have to go to pharmacophore, and here you have an option called search, screen and profile. Now I'll simply screen I'll go option for screen library. Here if you see, this is the pharmacophore. Already it has been taken by default, that is whatever the pharmacophore I have generated, that is 1SC7. By default it has taken, and this is the Q is my molecule, and I'll simply run. Once I do that, what happens? This Q will go and fit into the pharmacophore and tells me how well the complementary binding, whether there are any complementary binding with the receptor pocket, and in what iterations which is having in the space with the receptor pocket will also be displayed. Now if you see the result, 1 out of 1 molecule mapped into the pharmacophore. That means whatever our pharmacophore we designed in the receptor pocket, that exactly matching with the ligand. So this is the best results. You go for view best results. Once you click that, so this is your pharmacophore. You simply click the Q molecule. See how well it is fitting into the ligand pocket. So this is the fundamental information. You need to know the best fit model of all fit. You can even change the results pattern. You can even see how well the molecule is uh, designed or fit into the active pocket. So this is the fundamentals of QSAR modeling. Especially this is called as e-pharmacophore guided modeling of your design. Suppose if you are designing any molecules against such target, that is cancer or anything, you should aim your target at this level. Hope you understand. And this type of molecular docking studies using active site, that is QSAR based, is called as pharmacophore or e-pharmacophore or interactive pharmacophore design. Especially this type of docking is called as structure based drug design. Hope you like my lectures. Subscribe and share among your friends. Thank you. This is Dushyant Reddy signing off for today.